Hello guys, my name is Ali and welcome to our second video on the impulse conduction inside the heart. And please share our club Xmed education with your friends and subscribe to our channels. So let's start. So in this video we're gonna talk about how the current inside the heart go from one cell to another cell. Then we're gonna talk about the conduction velocity of this current system inside the heart, and then we're gonna see the effect of sympathetic and parasympathetic nervous system on the conduction velocity. And this is different from what we talked in our previous lecture, that we talked about the effect of sympathetic and parasympathetic effect on the SA nodes, which had the different mechanism than this one. And in the end we're gonna talk about the pathway of conduction system. So let's see. Impulse conduction inside the heart. So I want to first talk about this picture that you can see here. It's very beautiful that they show how this conduction system work inside the heart. So first, from the SA node, they go to the atrium and contract the atrium, and from different paths they go to the AV node and bundle of His, then bundle branches, then they go to the base of the heart via the Purkinje fibers, and then they go all the way in the wall between the atrium and ventricle. So how this exactly work in the molecular basis? As you see, these impulses are rhythmic and rapid. See, like they are in the rhythmic pattern, and they happen very rapidly. As you just need to put your hand on your heart and you see how rapid this system works. These impulses are made in a SA node, as we explained in our previous lecture, and these are like currents inside the electricity. So the mechanism is pretty same. And how they actually works, as I explained about the action potentials, these action potentials should go from cell to cell. This pattern need a gap junction, which is between the cells. Like for example, in the myocytes there is a gap junction between each cells, and current from one cell goes to another cell and cause the depolarization in the next cells. So this gap junction is an electrical synapse, and these surfaces that we talk about is intercalated discs of myocytes. And first you have to know that because the pulses start from the SA node, they contract the atrium first, then they go to the AV node, and then they contract the ventricles. So that's about the impulse conduction in the heart. And now we have to talk about the velocity. You know, everything is the velocity in this system, and this velocity depends on different factors. For example, the gap junction density. In the place that we want to talk, we have to know like what is the density, how many gap junction is between the cells. That's really important. Just imagine that you want to pass from like one pathway and there are some security stops. So if there is only one security stop, you have to wait more. If there are like 10 security stop, you don't need like in one row. If there is 10 sections, you don't need to wait that much. The second is the fiber diameter. Just imagine that you have one road which is just one car can pass, or you have a like bigger road that many cars can pass. So definitely if the road is larger in diameter, so they can more car can pass from that path and from that road. The third things is the tissue resistance, like how currents can pass through that tissue. Is this tissue is something that the resistance inside it is high, or it's not that high. And the magnitude of the depolarization current, so how strong is this current that wanna pass. If it's like something small, so they don't have a like very high speed, but like if the magnitude is pretty high, the speed gonna increase. And as you see here, there are some numbers that they show the velocity of conduction. Please don't mix this with the frequency of impulse generation in nodes and in the SA node and AV node that we explain in part 1 of these videos. So this is the velocity of the conduction, and this velocity is slow in the SA node and AV node. Why? As I explained, the number of gap junction are important, and in these two nodes the number are limited. So when the number is limited, the resistance goes up. As I explained, there is one, just imagine there is one like security control, or there are many security control in one place. Then the other thing that you need to know is the Purkinje fibers, the speed is really high because of two reasons. First of all, the diameter, look at this diameter here, they are pretty big, you know, like this 70 to 80 micrometer, which is the like large number. And the other thing is they have many gap junction. When they have many gap junction, the resistance goes down and the speed goes up. And this velocity of conduction control by sympathetic and parasympathetic system. So sympathetic can increase CAMP and parasympathetic decrease CAMP. And how they can do that? They increase the CAMP by GS as they activate like GS, like norepinephrine active GS increase the CAMP, and CAMP increase, they can affect on this velocity and they increase it. And parasympathetic, they affect on the M2 receptors, and by acetylcholine they decrease the CAMP by activating GI coupled. And this velocity control by positive, like this system that we talk, the velocity of conduction, means like positive and negative dromotropic effect. Please don't mix it up with the chronotropic effect that can relate it to the heart rate, and they can relate it to the SA node conduction. So if we affect somehow, like the sympathetic nervous system affect on the SA node and cause the increase the heart rate inside the SA node, the number of pulses will increase. When the number of pulses increase, the heart rate will increase. So we call it chronotropic effect, and it will increase. But like if they affect on the speed of this path, the dromotropic effect will be positive and negative. These two are different. So now we're gonna talk about the conduction pathway. In the conduction pathway, I already explained it in previous video and in this video, but like I'm gonna go in more detail here. So first we have SA node here on the right atrium, lateral to the inlet of the superior vena cava. Superior vena cava bring deoxygenated blood from the upper body to the right atrium, and SA node is close to that. So first from the SA node we have different pathways. Like one pathways goes to the left atrium, this called anterior interatrial band. Inter means like between the atrium. And they go to the left atrium, and we have three other pathways: anterior, middle, and posterior internodals, which means between two nodes, between SA node and AV node. They send the signals to AV node, but you have to know the speed inside the atrium is different. Sometimes these pulses go to the muscle myocardium of the atrium, and from the myocardium of the atrium they go to the AV node. But sometimes they can go through these bands that I explained. So if they go through the muscles, the speed is lower. But if they go through these pathways, the speed is higher, because like they are specialized for transferring these pulses. So that's why we have like two different speed. The speed, if they go just from the A myocyte is gonna be around 3 millisecond, and if they go from the these pathways, it can come up to like 1 millisecond per meter. The velocity will increase by passing through these bands. And why is it important? In some people, like when you're getting old, this pathway might not work very well, and the muscle can be the major mechanism. They can cause a problem. You're gonna study all this stuff in pathology. I just wanna give you some insight that how this work and how they can why they are important to know them. So then we go to the AV node. AV node from the posterior wall of the right atrium. They are in the posterior wall of the right atrium, and in the AV node we have some delay before the pulses start to going to the septum between the ventricles. They have some delays here. So then they send the signals, and this delay is helpful because, like as I mentioned, the speed are different, and this delay can help this pathway to go at the same time. And after that there is a bundle of his, and then bundle branches that they go to the left and right. It's really important that we have two bundled branches, because like when you study about the pathology of ECG, you see that we have right bundle branch blocks and left bundle branch blocks, that they have different mechanisms and they cause different problems. So it's important to know them. And we have Purkinje fibers. I already mentioned that Purkinje fibers has the largest fiber among all these fibers, and they definitely have higher velocity compared to others. And in the end, there is something that's really important to know. It's really important to know that when the contraction arrives to the muscle of the ventricles, they goes from the endocardial surface to the epicardial surface. Endocardial surface is here. So first the pulses goes here, then they go to the outside the cell. So it's like they go this way, And why is it important? The way, for example, you know, all of you guys probably had a ECG check, and you saw it in your physiology lab that they put some leads on your body, and they can check the some like ECG pattern of your heart, and we can see that because of this pattern, that they goes from inside the heart to the outside the heart. So that's how we can see it, that how they work. And this is important in case of, for example, myocardial infarction. When someone has a myocardial infarction, it means like some part of their heart doesn't receive blood, and that part will necrose. So we don't see this transmission from the inside to the outside. So in the ECG we see some difference because of that. So this is about the conduction system. I appreciate if you guys write a comment for me and like my videos and share it with your friends, and let me know how I can improve making these videos. Thank you very much, and see you next time.